Hello, I'm Thaovi and Nguyen, and today I'll be talking about chirality and structural isomers, impacts on oral health, how molecular structure shapes product effectiveness, and patient safety. The first thing I would like to ask is how do chiral molecules and structural isomers used in dental hygiene products affect their performance and patient safety? We'll first answer the impact on product performance. It can affect the taste and sensory experience. Many flavoring agents and sweeteners are chiral, like L-menthol, found in many everyday oral care products like toothpaste and mouthwash. It adds a minty flavor, freshness, and a cooling sensation. It can also do anti-carcinogenic effects. Chiral and structural differences can change how a compound interacts with oral bacteria, like Streptococcus mutans. An example is sugar alcohols. um, Sugar alcohols are shaped in a way that S mutants cannot easily metabolize, reducing acid production and lowering cavity risk. Then we talk about stability, solubility, and delivery. Molecules that differ in structure can dissolve differently or bind to surfaces differently. This can be how this affects and influence how well the product works, how long it lasts, and how safe it is. For example, it can be like toothpaste spread, one can be gritty and one can be smooth. And then here I have an example of L-menthol and D-menthol. As I said before, M-menthol provides the minty flavor, freshness, and cooling sensation, while D-menthol is a much weaker cooling effect. Now we're going to talk about how impact on patient safety. It can reduce the side effects. Many essential oils in toothpaste and mouthwash, such as menthol, carvone, and eucalyptus, are chiral. L-menthol provides the pleasant and cooling sensation, like I said before, while D-menthol is not as strong. It can also cause some irritation. Also, sugar alcohols like xylitol is well tolerated, but some stereoisomers or high doses of it can cause gastrointestinal upset, highlighting the importance of using the correct form. And toxin avoidance and metabolism. Fluoride containing products often include stabilizers or flavoring agents that are stereochemically specific. Some molecules are harmless in one stereochemical form but toxic in another. The inactive or mismatched form can build up in our bodies, and this can be like consuming xylitol again, where it's safe but the incorrect stereoisomer xylitol can be metabolized by oral bacteria. And here I have on the side xylitol and then its other incorrect stereoisomers that won't have the correct effect. And now I'll be talking about some examples of chiral compounds in oral care. The first thing we have again is L-menthol. It provides the minty cooling flavor in toothpaste, mouthwash, and gum. And then we have L-limonene. It is a citrus flavor in toothpaste and gum. And then we have R-carvone. It provides the spearmint flavor in gum and toothpaste. And I would like to note that their S form smells more like caraway, when their R form smells like spearmint. Then we have chiral resin intermediates. They can be in dental resins and stabilizers. The correct enantiomer of it is safe, but if the stereochemistry was wrong, it can be toxic in the oral cavity. Now we're going to talk about how structural differences change oral microbial activity. Oral microbes interact with molecules in the mouth every second. Even tiny changes in molecular structure change how bacteria behave because bacteria use specific enzymes to metabolize molecules. It can bind and form biofilms using specific surface receptors, and there can be bacterial toxins in signaling molecules, depends on the precise stereochemistry. So first, let's talk about chirality affects enzyme recognition. Bacterial enzymes are highly stereospecific. One stereoisomer may be easily metabolized, producing lactic acid, which lowers the pH, increasing the caries risk, while the other can be a mirror image molecule and may not be recognized. There can be no acid production, and bacteria will starve or slow down. Second, functional group placement affects fermentation. Changing the position of hydroxyl or carboxyl on a sugar or amino acid can alter whether bacteria can ferment it, change fermentation speed, and change the types of acids produced. Example can be the small structural changes determine whether bacteria produce lactic acid, acetic acid, ethanol, or no acid at all. Third is carbon chain length affects membrane penetration. Shorter molecules can diffuse into bacterial membranes or biofilms more easily, while longer or bulkier molecules diffuse more slowly, are broken down less efficiently, and can disrupt membrane fluidity. This changes the growth rate, biofilm density, and the bacterial communication. Fourth is double bonds or ring structures change binding behavior. Bacterial receptors interact differently with saturated versus unsaturated, cyclic or linear, or aromatic rings. These structural shifts influence adhesion to the enamel, biofilm architecture, or co-aggregation with other species. Fifth is structural stability changes micrometabolism. More stable molecules resist hydrolysis, oxidation, and bacterial degradation, while less stable or more reactive structures are that can break down faster, feed bacteria more easily, or increase metabolic acidity. So now moving on to our experiment, we are comparing two sugar alcohols for sweetness and bacterial growth inhibition. Our objective is to determine whether two sugar alcohols like xylitol and sorbitol differ in sweetness intensity, and second, ability to inhibit the growth of Streptococcus mutans. This is in order to evaluate whether these substances appear to possess anti-carcinogenic potential. Now let's talk a little bit about the background. Dental caries arise when acidogenic bacteria such as S. mutans ferment dietary sugars and produce acidic byproducts that lower the plaque pH. Sugar alcohols are commonly investigated as alternatives to sucrose because they offer sweetness similar to sugar. They poorly metabolize by carcinogenic bacteria, and some sugar alcohols have direct inhibitory effects on bacterial growth and metabolism. However, differences in structure, including the number of carbons and stereochemistry, cause sugar alcohols to behave differently in their oral environment. Dietary sugars are simple carbohydrates found naturally in foods or added during processing. They metabolize monosaccharides and disaccharides, the forms most easily metabolized by humans and oral bacteria. So first, I'll talk about xylitol. Xylitol is uptaked by the S mutants by the PTS system that recognizes xylitol because it is a 5-carbon sugar alcohol. Similar in structure to sugars it normally metabolizes like fructose. This allows xylitol to enter the bacterial cell. Once inside, xylitol is phosphorylated to xylitol 5-phosphate. The bacterium cannot metabolize xylitol 5-phosphate further, so no energy or biomass is produced from it. The bacterium must actively pump xylitol 5-phosphate out of the cell, and this uses the ATP and creates a metabolic stress. The bacterium wastes the energy trying to handle a molecule it cannot use. As a result, growth of the S mutant slows down, less acid is produced because metabolism is disrupted, and the biofilm formation is reduced, which helps prevent dental caries. Now let's talk about sorbitol. Sorbitol is a 6-carbon ring, and its bacterial system the bacterial system of S mutants does not recognize them, or as efficiently it can tolerate them without triggering stress. Once inside, they either are metabolized slowly, and this does not form toxic intermediates that wastes the energy compared to xylitol. As a result, little or no ATP is wasted, the bacterial growth is not strongly inhibited, and acid production continues. The key point is that the carbon number and structure determine whether the sugar alcohol is disrupted to bacteria. Wrong size or arrangement is less effective. And now we have the hypothesis for the experiment. The hypothesis is xylitol will inhibit more bacterial growth and produce a different sweetness concentration compared to sorbitol due to structural differences. And the null hypothesis is there will be no difference between xylitol and sorbitol in sweetness and or bacterial inhibition. So obviously I can't perform the experiment myself because I don't have the right materials or products. So I found 2 great research articles that performed the differences between the xylitol and sorbitol in bacterial inhibition, and also comparing xylitol and sorbitol with sucrose to see if the sweetness levels are the same. So this is the first one. You can take a look. And this is the second research paper I found. So we're going to talk the first table cited. Table 2 shows that xylitol reduced S mutants by 27% with p-value of 0.01, whereas sorbitol only reduced them by 3%. This means it's not significant. This selective inhibition is due to xylitol's structure, a 5 carbon, which is metabolized by bacteria into a toxic intermediate causing metabolic stress, while sorbitol, which is a 6 carbon sugar alcohol, is poorly metabolized. So, based on this table and its results from the research paper, um, the p-value for sorbitol is over 0.05. So that means it's not significant, and it will not reject the null hypothesis. So xylitol will inhibit more bacterial growth compared to sorbitol, because sorbitol has a negative 27% and has a p-value of 0.01. Next, we'll talk about Table 4, which is also cited. It shows that xylitol achieves sucrose-like sweetness at lower concentrations than sorbitol, reflecting the structural differences in carbon chain length and hydroxyl positioning that affect the interaction with sweet taste receptors. Together, these structural differences explain why xylitol is both more effective at inhibiting carcinogenic bacteria and perceived as sweeter than sorbitol. As we can see, let's look at sucrose, which is our comparing. We can see that at 5% concentration, it's at 5, and so on forth. And if you compare that with sorbitol and xylitol, xylitol has a closer concentration to sucrose compared to sorbitol. This means that xylitol is one of the better substitutions for sucrose, as the sweetness is around the same as sucrose compared to sorbitol. Now we ask ourselves, can these substances appear to possess anti-carcinogenic potential? My answer is yes. Xylitol appears to possess anti-carcinogenic potential while sorbitol is much less effective. Evidence from Bador et al. 2012 shows that xylitol gum reduced S mutant counts by 27% and a p-value of 0.01, whereas sorbitol gum only reduced counts by 3%, which is not significant. This effect is due to structural differences. Xylitol is a 5 carbon sugar alcohol and is taken up by carcinogenic bacteria and converted into a toxic intermediate that creates metabolic stress, inhibiting the growth and acid production, while sorbitol, a 6 carbon sugar alcohol, is poorly metabolized and does not induce some stress, so it has minimal anti-carcinogenic effects. Additionally, xylitol's structure configuration allows it to provide sweetness at lower concentrations than sorbitol, which supports its use in oral care products by enhancing taste while reducing bacterial activity. This shows that xylitol is both effective at inhibiting carcinogenic bacteria and functionally useful as a sweetener, confirming its anti-carcinogenic potential. And this is my conclusion. Again, it's the same thing I said. Xylitol's 5 carbon structure causes metabolic stress on bacteria. It interacts more efficiently with sweet taste receptors and demonstrates an anti-carcinogenic potential and functional sweetness, like in toothpaste and gum. And this is all backed up by the evidence that I recently stated before. And here are my references.